Hello, everyone, and welcome to today's Bytesize Talk. With us today is Luisa Santos from CRG, and today she's talking about another NF Core pipeline that was recently released, which is NF Core multiple sequence align. Off to you, Luisa.
Thank you. So today with the talk, I'd like to touch on a couple of things. So first, I want to briefly introduce this pipeline in what are the broader research perspectives that this pipeline really falls into. I'll touch on this one very briefly because I talked about this in the Nexus Summit. And most of the time, I will spend on explaining what this pipeline really can do for you now and how you can use it. So it will be quite technical. So this project started with the issue of tool selection in bioinformatics. And what I mean by this is that, you know, that in many different problems in bioinformatics, you many times have alternative solutions. So you have alternative methods that can produce alternative solutions. And the very one reason why we even talk about having alternative solutions is because none of them is the best one consistently for each dataset that we have. Otherwise, we would not even think about them as alternative solutions. This happens for a variety of reasons, but eventually the problem is that we keep having these alternative tools. And this is true in many cases. So I'm dropping here three, so protein folding, differential expression, and multiple sequence alignments, but this is very true for many others. I will focus today on multiple sequence alignments. And so really this project revolves around how to handle these alternative solutions. Do we completely solve this problem with this pipeline? We don't, but is it a step towards the right direction? I hope so, I believe so. So the very, very first thing that we started doing with this was to create at least a homogenized distribution of these tools. Now, so of course we picked NF Core modules. So we started just by creating a collection of NF Core modules for all of the alternative tools. And so we call this a method hub because basically they are all alternative solutions that basically produce the same thing. Like they all produce a multiple sequence alignments, but they use different algorithmic procedures. Now, of course, once we have the method hub, it's not functional per se. So we built NF Core multiple sequence align around this method hub to be able to first just deploy this method. But then we went slightly more ambitious and we thought, okay, it would be nice also to be able to combine these alternative solutions and also to compare them and evaluate them. In fact, we do see this pipeline as also purely a deployment framework if you want to use it as a deployment framework, but mostly the direction that we're going towards is making it as a benchmarking framework or a leaderboard place where you can really compare and evaluate and understand how these methods work. Now, what I will tell you today is about the Nova Icaria release, so all the parameter names and everything. And so because, just to catch you up on the naming scheme, so we work at CRG, and the beach in front of CRG is called Somorrostro, and now the naming scheme is all the beaches from Somorrostro on. So now we're at Nova Icaria, and hopefully we'll be at Bogatel soon. And so I'll first touch on the overview of the pipeline very briefly, and then I'll go step by step into each of these different steps. So the input that you will need to run this pipeline is either a FASTA file or a set of PDB structures. Then the very first thing that we do is to kind of try to understand how does the input look like. In fact, you could have, you know, longer sequences, shorter sequences, more similar sequences, more dissimilar sequences, and all of these can really affect how these tools work on these data sets. And so this is the first type of information that we collect. Then there is the guide tree computation. I will touch on this a little bit better in a sec. Then there is the star of the pipeline, so there is the alignment. And then if you want, you can compute a consensus solution. So this means that out of all of the alternative solutions that you may have computed in the alignment step, then you can combine the solution into one consensus one. Then there is the evaluation step, so you can evaluate your alignments. And then we provide different reporting and visualization of the summary, basically, that you have created during this pipeline. And so the main outputs of this pipeline are two. So one is really the alignment files, and then one is the summary and the reports that we create at the end. And I mean, of course, there are all the intermediate files if you want to. So, okay, now to get a little bit more in details. Okay, how does the input really look like? So I told you you can use, if you want, just purely a FASTA file, or you can use PDB structures. So how you would provide the PDB structures currently, you provide them within a folder. So really you provide the path to the folder where you have all your PDB structures stored. And why would you want to have both? You may just want to run both sequence-based and structure-based aligners, or you may even want to run a sequence-based aligners, but run a structural evaluation on it. So that's why you may want also to have PDB structures. Now, what if you have the PDB structures and you want to run a sequence-based aligner? Then in the pipeline, we can extract the FASTA file from the PDBs. So this is also supported. What if you only have the PDBs for certain structures, for certain samples, and not all of them? The ones that do not have the PDB structures, of course, will not be able to run the PDB-based either evaluations or alignments. So they will just be skipped, but the rest will run with the structures. Now, how you prepare the input file. So we have the very typical sample sheet as input. So you will have your identifier and your FASTA file. Then we have another column, which is the references column. So what is a reference? A reference, it's a reference alignment. So it's really a multiple sequence alignment. This could be technically anything, any alignment you want, but usually how people use it is there is benchmark data sets. And yeah, there are reference alignments that are usually manually curated, and you can use them within, you can provide them as input so that in the evaluation, they can be used to evaluate the final solution of your tools. Then we have the optional data column. Here currently we just have structures. We called it optional data because really we envisioned this pipeline to take as input not only protein structures, but at some point using potentially other informations. That's why we kept the name vague. And so here how you pass it, as I was mentioning before, you pass it really in the form of a folder. So you have a folder and within it you have all your structures. And finally, we have the template. This is, we call it template because it is the T-Coffee template. It's used currently only by T-Coffee, which is a tool that we provide within the pipeline. And it is literally a mapping between the IDs that you have within your FASTA file and the PDB structures that you have provided. In case you do not provide this template and you want to use T-Coffee, this will be automatically generated for you, of course, under the assumption that the ID within your FASTA file is the same one, has the same name as the PDB name. Otherwise, we have no way to really map them. But you can provide other mappings in case you had different namings. And yeah, then we have another input, which is the toolsheet. The toolsheet looks like this. So you have 4 columns at the moment. You have the tree, the arguments for the tree, the aligner, and the arguments for the aligner. Now, each row corresponds to one parallel run. So it's to a combination of guide tree and aligner. Why do we have a toolsheet and don't we have only in the configuration file run FAMSA RAM cluster? For 2 reasons, for many reasons, but the main 2 ones is 1, we want to be able to test alternative combination of the different methods in parallel, so within the same run to run the benchmark. And 2, we want to be able also to run alternative combination of the arguments of the different tools. In fact, there are many tools that if you use different arguments, they really deploy completely different algorithms. So in the benchmark, you really want to compare them in the same run. So then concerning the input file summary. So this is a sub-workflow. So you can skip it completely if you don't want to have it with skip stats. Or you can activate or deactivate each of the individual summaries with the flags that I put that are here on the right. Currently, what we collect is the percentage of similarity of the sequences. So you're just collecting how similar are these sequences. So of course, if you have very, very similar sequences, your alignment is going to be probably easier. And if you have very dissimilar one, it's going to be trickier. We also just report, how many sequences and how long they are. And the same thing, usually when you have many, many sequences, it can be trickier. Like usually there are more difficult alignments. And then finally, if your structures have been produced with AlphaFold 2, then they also have the PLDDT. So it's a structural accuracy metric. And if if the files, the structural files that you have the PLDT, we can extract it to also use it to collect it for the final report. Of course, now we only have these three, but the idea is to keep expanding these sub-workflows and add other modules. And then we have the guide tree. So as I was mentioning before, so basically the guide tree is a data structure that defines the order in which to align the sequences. This is a data structure that is used by some of the MSA algorithms, like not all of them, some of them do, most of them actually so far. And so some of them can use it as input. Now, currently there are 3 packages that we have included in the pipeline that render guide trees. These are Clustalo, FAMSA, and MAFT. And depending on the different arguments that you use with these tools, they can produce different trees. So in the documentation here, I'm putting a screenshot. We've added the different trees and the different arguments that you can use. And then on the last 3 columns, it's more specification of which exact algorithm, like which exact tree is being built. Then we have the alignment. The alignment, we provide different type of aligners, and we categorize them based on their input. So we have the purely sequence-based aligners, so they only take sequences as input. We have the ones that take both structures and sequences, and the ones that only take structures. Here is again a screenshot from the documentation, and we also add on the right of each tool whether they do accept the guide tree or not. Just to specify, none of these tools has to have a guide tree, in the sense that if you do not provide it explicitly, the tool will render it by itself. So the fact that we do have this combination of trees and aligners is mostly for a kind of advanced user that really want to test and play with the combinations of these guide tree and aligners. Then the consensus. So then once you have these alternative solutions, you can compute a consensus multiple sequence aligners. This is done with the tool called tCoffee. Currently, it's the only one that is integrated in the pipeline, but ideally, if more become available, we will integrate them. And so this tool does, on one side, it just produces the consensus MSA, and on the other side, it just renders, it just can give you information on plots or visualization like the one here that I'm putting, how to inform kind of which regions are better aligned or are more consistently aligned among the different solutions. Finally, we also have different evaluations for the different MSAs. Again, this is a sub-workflow, and it can be completely skipped by using the skip eval flag. And again, you have the flags on the right to turn on and off each of the different evaluations. So we have a few of them that are reference-based. So as I was mentioning before during the sample sheet, if you do provide the reference MSAs, then you can calculate this metric, otherwise obviously not. Then we have some that just need the MSA as input. These are like the amount, like just the amount of gaps that you have in your multiple sequence alignment, or the consistency, the total consistency score, which is a metric, an evaluation metric provided by tCoffee. And then you may also we also provide structure-based evaluations. So this one is the IRMST. Now, we also do provide different reports and visualizations. So the first one is multi-QC. In there, currently, we have like a very big summary table. So here we are basically collecting everything that went through the pipeline. So from, obviously, which tree, which arguments, which aligner, and which arguments. But we also have all the metrics that we have used for the input, that we have collected about the input files, and all the evaluations. Then we also have a little shiny app that we use to explore interactively the results. And finally, if you do provide the structures, we also provide the visualization with FoldMason. FoldMason is a structural aligner that is also within the pipeline as an aligner. But if you provide the structure, if you provide the structure, then you can also use the visualization. So you can just have this visual interface and play around with it. And so now how can you use the pipeline? So We have envisioned the pipeline for two different type of users. On one side, you just have the deployer, so someone that wants to just deploy their favorite tool and then that's it. Or the developer or someone who really is interested in more about benchmarking, and then it's a little bit more advanced. So if you're just on the side of the deployer and you just want to deploy one tool, then you can use the easy deploy profile. So basically this turns off everything that is more on the benchmark side. So the statistics, the evaluation and the validation and everything. And I didn't mention at the beginning, but if you do not want to provide the sample sheet and the tool sheet, you also have the alternative of using directly the -6, -6 or aligner and tree to provide directly the structure for one run with one data set, one tool, and one tree. If you want, the tree is optional here as well, of course. So if you were to run this tool, then it would just run quickly a validation on your FASTA file, compute your tree, compute your aligner, and then finally in your output, you would have your alignment and your tree. Now, what if you have only protein structures as input? Well, then you can use the PDB's dir instead of the 6. And in this case, the pipeline would first extract the FASTA from the PDB files, do a validation on the FASTA file, then compute the guide tree. The reason why it is extracting the FASTA file is really because the guide tree runs on these FASTA files, like the guide tree is sequence-based. And then you would run the structure-based aligner fold Mason. And then your results again are the alignments and the tree. Yes, so instead, if you were on the side of the developer and you want to run it using the tool sheet and the sample sheet, then you will have to use the ---- input, the ---- tools. And really here you can activate basically all of the different modules that are in the pipeline. And what you would have finally is different, so you would have, as before, the folder of aligner and trees, where you have per dataset all of the alternative alignments and trees correspondingly. And then you have all of the different reports, so multi-QC, the Shiny app, and the visualization. And then you have three other folders. So you have the folders of the statistics, where you have all of the different statistics and a summary of them. The evaluation, so same thing, all of the different evaluation and a summary of them. And then you have the final summary, which basically merges together everything. So the statistics, the evaluation, which tools were run. We also add in there the version of the tools. We add there also the information from the trace file, so running times, memory usage. We also use the CO2 plugin. So we also have the CO2 consumption. And because eventually these are very important metrics for the final benchmark. So we're not only interested in the accuracy, but also yeah, in the computational feasibility really of these tools. So this is why we have everything in this final CSV. And yeah, so just to summarize, so basically I touched on what are method hubs or what we envision as method hubs as, and how we generally design this pipeline thinking about it more in the terms of a benchmarking platform. And then, and mostly, I went through NF-core multiple sequence align and showed you how to compute, combine, and evaluate multiple sequence alignments with it. And yeah, thank you for listening. And I mean, this was a very collaborative project, so many people helped. Specifically Jose, Leon, Ale, and Igor, and Julia, who's not in this picture yet. And yeah, thanks for listening, and I'm happy to take questions.
Thank you very much. So I have allowed now everyone to unmute themselves. So if there are any questions, please unmute yourself and ask your question. Maybe I start. I was wondering, you're showing this all for protein sequences, right? Can you also put in nucleic acids?
Technically now, yes, if you turn off the validation. But although actually probably it will run anyway. But yes, you can. Now we have designed it in theory we have designed it as a protein, as a pipeline to align proteins. Yet the tools, many of the tools actually have that are within the pipeline actually have a mode to align DNA. So as long as you change the parameters, it will run on DNA. Indeed, part of the stuff that I was telling about the optional data is input with the structures. we kept it as optional data because there is an idea of going forward into kind of DNA. And potentially that's where it might become interesting to add different sources of information on DNA, because then, of course, you cannot have protein structures, but then you may have something different. You may add other layers of omics. And that's an area that we would like this pipeline to be able to explore. But currently, let's say that officially it's for protein sequences, but you can use it also for DNA.
Okay, cool. We have a question from Matthias. Very important. Are there plans for a pipeline tube map?
A pipeline?
A pipeline tube map, when you have the lines connecting the different steps.
So we had it. I really had one, and there was a tough decision to remove it. The thing is that so probably not, but because to make it technically correct, then because everything could run in parallel, then you would have to have so many tubes that then it becomes complicated. So then it would become either nice or maybe not super technically correct. Or if someone has ideas on how to draw it nicely, I'm happy to do it. But we tried, we had a poster with it, and we had to remove it in the end. Yeah.
Maybe we have to forward this question to James, who's our master of tube maps. Another very important question. Are you visiting each beach before the release?
Yes, actually, I mean, we visited all of them, but let's see. Well, now they're very close. All of them, I visited them, but when it goes towards Costa Brava, I'll be happy to also go for excursions.
Okay. Are there more questions from the audience? So I have yet another question. You say yourself that choosing the right tool is very difficult at times. Do you have any guidelines on when to choose which tool? Like, is there any documentation on that available? Maybe not specifically from your pipeline, but from somewhere, because it's tricky.
It is very tricky. There is a paper. How do I put it now? Well, no, there is a paper that kind of started having kind of decision tree on how to, like, depending on how many sequences you have and everything, but something, so it would give you some recommendation on this, but then something super systematic there is not, and this is the problem that kind of you're trying to go after. And what would be nice at some point, of course, would be this came out of a recommendation of someone or a suggestion of someone in the summit talk that they were wondering whether this could be used, like this pipeline could be used for the generation of training data, for then being able to actually provide recommendation, because eventually what you have, you have information about your input data, like how your data set looks like, and your information how well are these tools performing. So technically you could be able, given a certain input data, to say, okay, this one tool would perform a bit better or not. We are thinking about this, but there is nothing done yet. So it would be nice to move this towards that direction as well.
Yeah, if you happen to have a link to that paper, I can put it underneath the bytesize topic.
Yes, I will look for it. I don't have it on top of my head, but there is something, yeah.
Perfect. Cool. Are there more questions? It seems that is all the questions we have for today. I guess if someone has another question, they can contact you.
Yeah, sure.
Or otherwise write in the Multiple Sequence Align Slack channel. I'm sure there you will find help. Thank you so much for your time today, and also thanks to everyone in the audience. And just a little announcement, very little announcement. We will not have another bite-sized talk until the 1st of April. It's not a joke. But unfortunately, next week's bite size we had to cancel, and then it's too close to the hackathon, so we will have a short break and we'll be back in April. So thank you very much, everyone.
Thank you.
Bye-bye.